symposium. And we will be recording the symposium today. That's so that we can kind of share it in the future. We are excited to all come together and to come together with the people in the ALS community. And we have a lot of people who are sort of still joining now, so maybe we'll give it just a minute as people kind of get connected to this. And then we'll do a few introductions and we'll get started with our program. So maybe just a couple of minutes. I still see a lot of people joining, so we'll let that go. All right, so we about once a year, sometimes twice a year, come together to just sort of try to have an event where we can talk about some of the exciting things that are happening in ALS research and also in the clinic, and try to sort of come together to answer questions and sort of hear what people are thinking. And so we want to do that today. We have a full program. I think before we do the program, I would love to just maybe have everybody just give a quick introduction. You can see people's names in their little Zoom square, but it'd be nice to just hear from everybody. What I might do is just go through and kind of list people's names so that we're organized. So Merit, would you like to start?
Oh, hi. Welcome, everybody. Merit Sukovich. I'm director of the Healey Center and an ALS neurologist and trialist. Happy to be here.
Thanks. Mark?
Sure. I'm Mark Garay. I'm an instructor of neurology at the Healey Center at Mass General Hospital, and somewhat merit, I'm an ALS physician and clinical trialist, in my case, focusing a little bit on familial forms of ALS.
I need to be more specific with my names, but that was right. That was Suma Babu.
Hi, everyone. Welcome to this session. I'm Suma Babu. I'm an ALS neurologist, a clinical trialist, and I also lead the neuroimaging program for ALS at MGH.
Sabrina Paganoni.
Hi, everyone. This is Sabrina Paganoni. I am an ALS physician and clinical trialist.
Doreen Ho.
Good afternoon, everyone. It's a pleasure to be here. My name is Doreen Ho. I am a clinical trialist and ALS physician.
Jen Scalia.
Hi, I'm Jen Scalia. I'm a nurse practitioner, and I help to co-direct the ALS clinic at Mass General.
Mark Albers, the source of the trouble here.
Good afternoon, everyone. My name is Mark Albers. I'm a neurologist at Mass General Hospital and run a lab related to neurodegeneration, and excited to share some of our results today.
Amanda Maidman.
Hi, my name is Amanda Maidman. I'm the program manager for the NCRI, and my function is to put together organizational education meetings such as this.
And Juliana Araujo. Oh, you're on mute, Juliana.
First one to do that. Sorry about that. I'm Juliana. I am the communication specialist here at the Healey AMG Center. Thank you for being here today.
Thanks. And I'm James Berry. So I'm one of the ALS clinicians and clinical researchers, and I lead the division of ALS at MGH. I look forward to these events every year. I think this is a great time for us to kind of look back at kind of what the year preceding has held, all of the advances. And while that's always been an exciting thing to do, over the last couple of years, it's been really remarkable how much changes one year to the next. And this year is certainly no exception. I would say more than ever before, we have new things to talk about, new things to share, and new things for you to ask questions about. So we are, at the Healey Center and in the clinic, we are incredibly excited about the progress that we're making. We know that there is more progress to be made and that we need to go faster. And we take that very seriously. We think of our clinical care as the cornerstone of what we do, and as research as really a part of that care, because we know that we need to make more progress. I think all of us take that very seriously, and you'll hear about some of that today. With that, I'm going to hand over to Merit Sukovich to give us sort of an overview of the landscape of how things are changing in ALS research and trials.
Thank you, James, and thank you, Juliana, for organizing and putting up the slides. We are at like a incredible time right now with new drugs on the market and so many trials going on, and it's almost impossible to update in a short amount of time. All the good things that are going on, but I'll do my best. So next slide, please. As I know everybody here on the call knows that this, although considered a rare disease, it really isn't. Lifetime risk of getting ALS is somewhere between 1 in 350 people and 1 in 440. And it gets more common as people get older, even though the average age is 55. And, we see people in the teens to the 90s who get this. A lot of the work has been on understanding both the type of ALS that's caused by changes in your DNA and genetic mutations, but also a lot of science going on in the other 90 or 80% of people who do not have a known gene mutation. There are so many people working on ALS. When I started in the field, maybe there was 100, and we all saw each other a lot, and there wasn't a lot of funding around, and it was an amazing community that worked very hard. But it's so different now. It's still an amazing community, but it's thousands of people and hundreds of companies and a really engaged advocacy patient population, a partnership between everybody working on this, and it's really coming to fruition now with more insight into the biology, more treatments and development, and more things already on the market. So it's super hopeful and exciting. The other thing I'd say is that we still are looking for drugs that can slow and ultimately stop the progression, but there's also efforts now, as you'll hear later, to try to prevent the onset in the first place, and also to repair function once lost. So that is also gives me a lot of hope and excitement. Next slide, please. So things are moving fast, and I will say that it's a little harder now to know which is the best trial for a particular person, and the team here at Mass General wants to really work hard with our patients, and also help other sites and other people know what their options are to make the best options. And we can't develop treatments without people participating in the trials, and so we're very grateful for that. Because of that participation, we now have 4 disease-modifying medications on the market. 3 that start with R: Riluzole, Radicava, and Relyvrio, with the Relyvrio being the most recent one in September. For all forms of ALS, and Radicava, the oral, a little earlier this year in 2022. But really just a week ago, the FDA approved for one of the genetic forms of ALS, a drug called, I think it's pronounced Qalsody, I'm not positive, but we call it tofersen, and this is a gene therapy for people with SOD1 mutations. And it was the first drug in ALS approved based using a mechanism called accelerated approval, which is approval on a change in the biomarker. And that's important because fields that can develop those type of biomarkers that change in a short time and predict clinical effect see the numbers of drugs approved go up exponentially. So that there's many reasons to be very excited about that recent approval. Besides those drugs, there are medications that help with symptoms and supportive care. And we know from many, many studies that people do much better who get that type of multidisciplinary care, like what you'll hear about that the Healey Center offers. There are many, many trial options. I'll go through some of them. But again, we want to work individually with people to try to pick what might be the best option for them. There's never been a time like this where everybody's really rowing in sync. The foundations, the people and the families with ALS, the clinicians, the scientists, the companies, all really focus on trying to accelerate therapy development. Last I counted, there were more than 300 companies now working in ALS. We started a trial network in 1995, the NEALS consortium, but now there's a European one, TriCals, there's a Pan-Asian one, PACT ALS, and all these networks are working together so that we can, not necessarily test the same drugs, but test different ones and share information, share knowledge about how to do trials. As you'll hear from Sabrina, we launched the first platform trial, which is a way to accelerate therapy development in 2020. And we have a lot of really exciting therapies that are being tested in that platform. Next slide, please. So I mentioned NEALS. This is an organization that started in the Northeast ALS, but really now has 147 centers, largely in North America, but also in South America and the Middle East and parts of Europe. And here, anyone who studies, touches, cares for people with ALS is part of this consortium. And the goal of this consortium is to lower the barriers to get access to treatments, do trials better, and advance knowledge about the illness. Next slide, please. So this group has a great relationship with many of you probably on this call, as well as other leaders really out in the community, to try to keep improving what we do. And one of our principles is that we don't nobody has time in ALS. We need to work efficiently and fast. So everything we do is shared, including the samples from all the studies, the clinical data. We try to speed up how we can get studies started. We train people so that no matter where somebody lives, there could be a center near you. And we innovate on how to do trials. And we talk through our science advisory board, any company that has a interest in ALS, we meet with them and help them with both their preclinical studies as well as their trial design. Again, with the goal of lowering barriers and speeding up progress. Next slide, please. So this is a list of current trials that are in what we call later stage, phase 2 or 3, where they're looking at efficacy. And this might not be completely up to date. I try to update it, but there's so much going on globally. So just to say on the left here, the ones in red are trials that have recently finished and were negative. And so we always learn no matter what, but obviously we want drugs to work. And the ones in blue are finished and have, depending on the one, they have some positive data and are still under development. And then the ones in the middle are active trials. So there's in Europe, for example, 2 studies of everyday edaravone versus placebo in different formulations that are ongoing. There's a repeat study in Europe of Relyvrio, because in Europe they have not approved Relyvrio, even though in the United States they have approved it. So there's a repeat study going on there to meet the European requirements. There's phase 3 trials of abutalas and masitinib, which are 2 drugs that work on different parts of the inflammatory pathway. The next one, whose name is unpronounceable, lenzumestrocel, is a stem cell trial by CoreStem as a phase 3 trial. Is being done in South Korea that the FDA has agreed to accept if it's positive. The SAR, I'm going to tell you a little bit more. That's by Sanofi, and is a drug that works on inflammation and cell death pathways. And there's a few at the bottom here that are really targeted towards smaller populations. For example, a gene therapy for people with a particular mutation in FUS. There's a prevention trial now with the same drug that's on the market for SOD1, giving it to people who carry the gene but are not yet affected. And then because of all this activity, we really decided that it didn't make sense to keep testing drugs one at a time, and that we should really learn from other fields like cancer, where they have used more efficient trial designs like the platform trial. And you'll hear from Dr. Paganoni about that. Next slide. Just wanted to show you a little bit about one of the trials. I just picked one that I thought maybe has not had that much information out there, and that we're actively enrolling. And this is called the HEALEY trial, led by Sanofi. And actually led by someone, Dr. Atassi, who sees patients with us and had been at Mass General for 10 years and then left to go to industry, and is leading their ALS trial. Next slide. This is a drug that works on something called RIPK1. So it inhibits something called RIPK1. And when it does that, it can block inflammation and block steps that lead to cell death. They did a phase 1 trial, which is typical, where you look to see if what dose to give and is it safe, and is there anything you need to follow. And they did that in people without any ALS, and what they call healthy controls. And they were able to show that they could get into the brain, they hit their targets, and it was safe. And now they started a phase 2 trial, which is a global study. Next slide. For the mechanism, it works on 2 pathways, really. One, by blocking this RIPK1, it inhibits inflammatory changes that can lead the motor neurons to die or to be injured. Next, please. It can also work on steps that that are activated in people with ALS that lead to the cells dying. So it really has a two-pronged attack on inflammation and cell death pathways. Next slide. So the design is one more, please, is a 24-week study. In the first part, people are randomized 2-to-1 to active drug versus placebo for 24 weeks. And there's 261 people in this study. And then after 24 weeks, everybody goes on the active drug and they stay on that until the results of the first 24 weeks are known. This study is enrolling very well and is anticipated to finish enrollment perhaps in August or July. So Mass General is a site in this, and they are recruiting for this study. This is an oral drug. Next slide, please. So I want to, that was a brief overview of the landscape. It's quite exciting. It's very different and it's very hopeful. And our center really believes from day one that we're only going to cure this illness with collaborations and partnership and thinking always about speed and innovating about how we can do better. And I'm very excited to see really some huge progress this year with 3 drugs approved in less than a year. And so I feel like we're in a huge momentum. Look forward to any questions. Thank you.
Thanks so much, Merritt. I mean, it's amazing to me, even having been part of it for many years, it's amazing to me how much is going on. How do people, I don't see any questions right now, but how do people make decisions about what trial's right for them? I mean, how do you think about that?
I think it's harder than it used to be. We used to only have maybe 1 or 2 trials. I think it's really important to gather the information about it. And for that reason, talking to your neurologist, obviously, but also we have an amazing woman, Judy Carey, who's our research access nurse, of spending some time with her, and she'll do this by Zoom or one-on-one, to go through the trials, the science, maybe the inclusion, exclusion criteria, how long it is, what's involved, so that you can make an educated decision. And I do think it takes a little time, maybe 1 or 2 times talking to your physician and Judy. Sometimes it's obvious, like if somebody has a gene mutation and there's a trial for that, we usually, say to go for that because that's right on target. For people without the gene mutation, I think it's really looking at the level of science and what's involved.
That's great. I mean, I think that's often a question that we're, talking with people about in our clinic visits and, in between clinic visits. And and there is no perfect answer. There's sort of the best answer for that person. Maybe is the right thing. But we do hope that people have an opportunity. And if anybody out there feels like they haven't had an opportunity to participate in research or don't know about research, we're all happy to answer those questions. All right. So with that setting the stage, I think what we can do is move on to Sabrina Paganoni, who is going to talk about the HEALEY Platform Trial, which is actually kind of devised and designed with Merritt as the PI and Sabrina as the co-PI to address this issue of so many drugs that we have available that may be helpful in slowing or halting progress in ALS. And so Sabrina, thank you.
Thank you so much. Yes, you said it right. That's exactly what we're trying to do. We're trying to work together to develop new treatments for ALS and do so faster. As you heard from Dr. Sukovich and Dr. Berry, really there's a lot of progress in ALS right now. We have drugs in development. We have many more that we ever had before. And so the reason we came together in 2020 to do something different like the platform trial was exactly to try to address the problem, which is a good problem to have, that now we have so many drugs in development that we want to make sure that we have a new way to test them faster. So let me just show you a 30-second video clip to explain how the platform trial is. Essentially, it's a big infrastructure. We're working together to test drugs, and this video tells you how.
The fight against ALS. The first ever ALS platform trial, designed to accelerate the development of effective and breakthrough treatments for people with ALS. So what's different about the HEALEY ALS platform trial? Instead of testing just one drug, the HEALEY ALS Platform Trial evaluates multiple treatments at the same time. This means more opportunities to find groundbreaking therapies in a faster time frame. Imagine having to build a new dock every time a different ship came to port with new treatment cargo. This is how traditional trials work. In platform trials, there is one dock to receive all the cargo and connect the treatments to their final destinations.
So this is what we did in 2020. We built that dock, that central destination where all the treatment cargoes really can work together and really be deployed and efficiently tested. And we continue to grow. We've been continuing to grow since then. In 2020, we started testing 3 drugs. Each drug is called a regimen in our trial. And then we continued to grow despite the pandemic and thanks to all the efforts and participation from so many in the community and so many companies working together. So we added a 4th drug in 2021, a 5th drug in 2022. And what I want to spend a little bit more time talking about today is what is our plans for 2023. Right now, we're very excited to be testing drugs F and G, and I'll tell you a little bit more about that. But again, just to explain how the new drugs sort of fit within the larger context. I just wanted to share that we built this common protocol and shared infrastructure to test multiple drugs. This is an efficient method because we have a single governance structure and we can use it multiple times to enroll hundreds of participants. Now we are over 1,000 participants at several sites in the country. So this is coordinated from MGH in collaboration with many partners, and we are excited to also offer the same opportunities to people from from around the country because we have over 70 enrolling sites. And I want to explain what we're also trying to do sort of scientifically. We're really trying to test multiple different mechanisms because the reality is that in different people, there may be multiple mechanisms that cause ALS. And so each drug has been testing different parts of the system. And by doing so, we're also able to learn a lot about the best targets for ALS. Again, we have many companies, many drugs in development, and we really want to test them one by one systematically to make progress. So the first couple of regimens A and B, 2 of the original group, were testing drugs that target neuroinflammation. And the 3rd drug tested energy production. The 4th drug is a sigma-1 receptor agonist, which we know is implied in ALS. And now the 5th drug was targeting autophagy. And now in 2023, we're launching 2 new regimens F and G. They are different molecules made by different companies, but they both target the same mechanism called the integrated stress response, which was recently shown to be potentially very important in the origin of ALS. So far, we have been testing drugs again efficiently. The platform is really an infrastructure to make progress fast and to also provide clear answers so that we know what to do with each single drug. In other words, we want to make go, no-go decisions fast so that we can make progress faster. The first 2 drugs, unfortunately, didn't have a positive outcome. And while disappointing, that makes that we were rapidly able to tell that they don't work. And therefore, we can refocus our resources and attention to other drugs. The regimen C and D, so drugs number 3 and 4, had some positive findings from the platform trial, so much so that now the respective companies can bring them forward to phase 3. So this was a success from our point of view because we really want to be able to again make go, no-go decisions quickly and only move the most promising drugs forward to the next phase of development. The 5th drug, Trialose, is still being tested, so we don't have the results. We expect the results by the end of the year, and we'll be sure to share all of this with you. And now let me turn to regimens F and G. Regimen F already started enrolling participants, and regimen G is very close to that. So we are very excited that we will have two drugs enrolling in the platform, including MGH very soon. Again, right now we started with F and we'll add G very soon. So whether it's F or G or any other letter, really, the concept is the same. We have a placebo-controlled trial, which we kept as short as possible to 6 months, followed by an active treatment extension. So what it means that from the point of view of a person with ALS who decides to participate in research, and in particular participate in the platform trial, there is an initial 6-month period where that individual will be assigned to either active or placebo, so that we can tell if the drug is effective for the treatment of ALS. And then we have a longer period active treatment extension where all participants, regardless of whether they were on active or placebo to start with, can receive active treatment long term. That second part is also very important to be able to tell if the drug is safe and effective. So essentially we have a number of different outcomes from the study. Each period is important, has different goals, and together both the placebo-controlled period and the active treatment extension period allow us to know if the drug has an effect on disease severity. In other words, does it slow down the progression of ALS? And also we can tell if the drug has an effect on other metrics, such as respiratory function, muscle strength, and a number of biomarkers, which we know are so important for us to learn more about ALS and really make progress. So F and G will follow the same general principle. And again, that's kind of the general schema. So that's what to expect when enrolling in the trial. Right now, we're enrolling for F. I borrowed a couple of slides from their webinar that they gave us to explain how their drug works. Again, it targets the integrated stress response, which is a pathway that's really of great interest right now in ALS. In fact, there are other drugs, such as the drug in regimen G, targeting the same pathway, although in different ways. And so essentially what happens in a nutshell is that when cells are healthy and there's no stress, sort of the cell function progresses normally. But when there is stress, the integrated stress response gets activated. And when this is only temporary, the cell can recover. But when there is chronic stress, as in ALS, what happens over time is that there is a toxic buildup of proteins that damage the cell function and eventually lead to the loss of motor neurons, which are the cells that are so important and whose loss causes ALS. So we're really trying to get to the core of these and try with this new drug to directly address this chronic stress and persistent activation of the integrated stress response, and ultimately, hopefully, trying to reduce this buildup of proteins and stress granules and protein aggregates that seem to be central to the origin of ALS. And so with regimen F in particular, this is a drug that was developed by Calico Labs in collaboration with AbbVie. That's regimen F in the platform trial, and we're enrolling for it right now, including at MGH. And if you want to have more information about this specific drug, we had a great webinar that was recorded. I'm sharing here sort of the link to the recording and the QR code so that, if you want to have more information about it, you can go back to that. Right now, we're also enrolling for this at 46 sites across the country. So in case you're not from the MGH area, you can always find other sites, and we continue to activate more sites. And certainly we want this short talk to really just be the beginning of a conversation for anyone who is interested in coordinating their care and research with us, and really considering participation in the platform trial. In addition to the resources that were mentioned earlier, obviously we have Judy Carey at MGH for our clinic, and in general for the platform trial, we have another similar research access group called the Patient Navigation Team. That's a central resource for information. So we provide information every week on Thursdays, and also there's information that can go out via the ALS link, and you can always call our patient navigators if you have questions about the trial, and I'm sharing all the information on the slide. So I just want to conclude, also share my email for anyone who may want to ask me questions after the webinar. And as always, we are incredibly grateful to everyone who's participating in this trial. So this trial is really meant to accelerate progress in ALS. And so many of you have either participated or contributed to supporting research in so many different ways. And so we just want to thank you, and we'll continue to test more drugs and really try to do this as efficiently as possible so that we can make more treatments available for all our patients.
Sabrina, thank you so much. A wonderful overview. it changes every time we hear about it because things are evolving so fast with the trial, which is, I think, one of the most amazing things about the trial. It can change with the science, it can grow, it can adapt. There's a really interesting question from Lynn here, which is kind of building on what's being done in the platform trial, which is testing things in parallel. Is there an opportunity to study more than one compound at the same time, perhaps one that addresses one sort of problem in the cell, as you showed, and one that's addressing another one?
Yeah, I completely agree. That would be fantastic. And I do think it's the way to go, in the sense that we probably need more cocktails for people with ALS, because we know that in different people, many different mechanisms may be activated. So perhaps only addressing one at a time is not enough. But that's actually, in a way, what we are already doing in the platform trial, because we are allowing every participant to take the standard of care medications. So, for example, Riluzole, Radicava, and Reliviro. And Reliviro itself actually is a combination of two compounds. So it's three, but really you get four, I guess. And so with those three background treatments kind of in the background, we're adding compounds to it. Now, I would say that we can potentially even test two new drugs at the same time. That's been done actually in cancer with the platform trial approach. I think that for now, for our initial sort of version of the platform trial, we are layering those new compounds one at a time on top of the three that are already available. But we can certainly continue to evolve and really consider, including two new drugs at the same time. That's a great question.
It is a great question. And I'll talk a little bit more about the approved drugs after our break. I think I'll underline the fact that we are kind of doing some combination therapy. And the other thing I'll say is that we just had the Target ALS conference, and there were a lot of sort of side conversations about preclinical work, that is, work that's going on in animal models of the disease, testing combinations of therapies, and how that gets complex. But really, there is a way forward for that from a regulatory perspective and a science perspective. And so I think that may be something we see more of, starting with the Amlex compound. Thank you so much, Sabrina.
Thank you so much.
All right. So we're going to keep moving along in our program. And next, we're going to have Mark Albers, who is the first part of the Mark duo here, is going to give a talk about neurodegenerative Alzheimer's disease and amyotrophic lateral sclerosis, or NADALS basket trial. And I will just say that this comes out of, so we have a meeting that we call the Translational ALS Science Meeting that happens once a month, and we get together bench researchers and clinical researchers and clinicians, and we talk about the science that's going on at every level. And the first time that I heard about Mark Albers' work was there at one of those meetings, and it's really come to fruition from an idea that was shared at a meeting where we think about some of the most innovative science to now a trial that's going on, and it's been just a joy to have any part in that. So, Mark, we're incredibly excited to have you here.
Well, thank you so much, and it's a joy to be here, and thank you everyone for joining. I'm going to tell you about our efforts about drug repurposing in ALS. We're using drugs that are already available. In particular, what we affectionately call it the basket trial, a trial that's a repurposed drug for patients with ALS as well as Alzheimer's disease. And these are my disclosures. The most important thing is that I'm going to be talking about an FDA-approved drug, but in an experimental context. This is not meant to be any clinical recommendation. So what is drug repurposing? That's when you take FDA-approved drugs for one disease and see if they can be helpful or work in a different disease. And as an example, there's a rheumatoid arthritis drug called baricitinib that was in the middle of the pandemic, was found to help people with COVID, particularly people that were hospitalized with COVID. It's an anti-inflammatory, and it reduced the chances that people moved into the ICU or needed intubation for those COVID infection. So that's one example. Why is drug repurposing interesting? A couple reasons. One is that the actual target of the drug may be active in more than one disease, and we're talking about inflammation in ALS, lots of different types of inflammation. And rheumatoid arthritis, of course, is an inflammatory disease. And so it could be an on-target mechanism. However, it turns out most FDA-approved drugs have more than one target. And so what you end up doing is not playing single notes on a piano, but you're actually playing chords. That the way the drugs work is inside the cell, they hit multiple targets and change multiple pathways. And so one drug might be a way to treat a different disease through one of the other targets. And you can turn it around and say, how can we use drugs to teach us about disease mechanisms? And so that's some of the work that we've been doing in neurodegeneration and ALS in particular. And so using this idea of a chord, we're trying to find new drug chords for ALS. And so one approach that we've used is there's a protein called 43 or TDP43, but I just call it 43. It's normally in the nucleus of cells, including motor neurons. If it comes out of the nucleus, then one of its roles is that it helps to suppress the parts of the DNA that are not used, parts of our genome that are not used to make proteins. And so when it comes out, it's like a Tupperware. Its function is like Tupperware. It keeps things content. And when it comes out of the nucleus, then you get more and more junk coming out. And when some of that comes out of the nucleus with it can mimic and pretend, makes the cell think it got infected by a virus. And so it then mounts a real anti-inflammatory effect. Sorry.
I apologize. I just wanted to jump in. There's something funny about your slides are sort of blinking for us, and I don't.
Oh, okay.
I was.
Let me stop the. How about this then?
Yeah, I wonder
if I don't do that. Okay.
That looks fine. You might try even restarting. We could see if it's still blinking.
Sorry. Thank you for letting me know.
Yeah. No, I'm sorry. We were trying to see if there was another way to fix it without breaking in. No, it's something about sharing this way. So yeah, it's better to go back to the other way.
Okay, so let me see if, so basically this is a brain from a patient, an ALS patient at Mass General, who donated their brain for research purposes, which is incredibly valuable. And what you're seeing is that there's an overlap. When the 43 protein comes out of the nucleus, the junk actually associates with it, and then it tricks the cell into thinking it was infected by a virus, even though it came from the genome itself, and mounts this strong anti-inflammatory pathway. So how do we know that? Well, we were able to recapitulate that in cells growing in culture. And so what you see here are in green are human neural cells, and in red is we took the junk, if you will, and made it red. And then we could add it to the cells themselves. And I can, let's see. Yeah, so the movie, I'll play the movie. So this is a movie over 48 hours. And what you see is that when the red material gets into the neural cells, it surrounds the middle of the cell, and then the cells die. And so this, although we see this in ALS, we also see the same mechanism in Alzheimer's disease. And with this particular phenomena, we were now in a position to screen drugs. And so by having a way to recapitulate what we're seeing in the human brains in culture, that allowed us to ask what drugs might block this. And so we looked at anti-inflammatory drugs, and we found 3 anti-inflammatory drugs that are in the same family. They're called JAK inhibitors. And they rescue the inflammation and they rescue the neuronal death in a dose-dependent manner. And one of them is baricitinib, the drug that I was telling you about that was helpful for COVID. It also seems to be important or relevant in this particular pathway. And so that's very interesting, but then how are we going to identify the patients that might have this type of inflammation? Going back to the discussion earlier about combination therapies and the complexity of the disease. And so we've been working with Mark Gray, James, and many others in the Niels and the Healey Center to look at the spinal fluids that patients have contributed and research participants have contributed. And we found some, including this particular inflammatory biomarker, which is called CCL2. And so with that, then we have shown that baricitinib knocks down CCL2 in the cell culture and in the mice dramatically. And so we're using this as a biomarker in the basket trial. And so just a little bit more about baricitinib. It's a pill. You take it once a day. You can use it through G-tubes as well. It's FDA approved for rheumatoid arthritis and more recently for hair loss, inflammatory mediated hair loss, as well as being emergency use authorization for COVID. With as other anti-inflammatory drugs, it has risks that are associated with infections of the upper respiratory, so colds, nausea, and then reactivation of zoster or reactivation of cold sores. And so the basket trial is actively recruiting right now for patients with ALS, just as Sabrina was talking about with you can be on Riluzole, Radicava, and Relyvrio and be in this trial. Because this is the way the trial is structured is the first thing we do is check for each patient to see if they have this type of inflammation in their spinal fluid. And so if they come in, they get a spinal tap, within 2 to 3 weeks, you get the answer, yes or no. And if you do have this type of inflammation, then you're invited to stay in the trial. If you don't have this type of inflammation, then we tell you that you don't, and this is not the best drug for you. If you are invited to stay in the trial, you then get a second spinal tap 8 weeks apart. And so that allows us to look at the stability of the spinal tap in each individual. And then we start on the 2 milligram dose of the drug for 8 weeks, another spinal tap, and then the 4 milligram dose of the drug, and then a fourth spinal tap at that point. And so that gives us then windows, 8 weeks off drugs, 8 weeks at 2 milligrams, and 8 weeks at 4 milligrams, in order to see if the drug is working in a dose-dependent manner to reduce the inflammation and to reduce the markers of neurodegenerations, such as NFL. And so every patient who is enrolled in this trial does receive the drug, and it's an FDA-approved drug. We do have one ALS patient enrolled right now and one asymptomatic gene carrier. So people that have the genes, and you're going to hear more about this from Mark Ray, but people that have the genes that are put people at risk for ALS can also come into this trial. And so I'm happy to answer any questions. I just want to thank, as James was saying, I mean, there's an enormous team that's behind this, both in the lab and the clinical research team. I did not know anything. I take care of patients, Alzheimer's patients, but I didn't know anything about clinical research. And the James and the Healey Center have been incredibly helpful to bring this out of the lab and into patients in a very rapid manner. And want to thank their support for this effort.
Thank you so much, Mark. This is really, it's so exciting. It's a little hard to hear that the drug has approval for hair loss before we, just as we're getting tested for ALS. But I think this has been, a journey to get this come together. And I think, just to be frank, we have to bring funding together. We have to get the trial design together, and the science has to come together all kind of at the same time. And so really excited to get this going. I'm gonna take a little bit of moderator initiative here, Mark. I think some of the science that led to this is so compelling as just a story. I would love you to tell just a little bit about how you kind of came to find, just at a very high level, the kind of the process of coming to find that this sort of inflammation was happening in cells, and what that, how that you were working with sort of a postdoc, and how that model sort of guided you in ways that you didn't expect.
No, absolutely. I mean, my clinical specialty is Alzheimer's, and this all started in a mouse model that was an Alzheimer's mouse model, but we had this peculiar genomic event that happened, and we got this inflammatory response, and we kept following it. And it turns out that once we understood what was going on in the mouse model, we then asked, is it happening in patients, in human brains? And so then when we saw that it was quite interesting in Alzheimer's. And then with the science advances, and particularly in the C9orf72 gene, one of the most common mutation associated with ALS, we saw that there was hallmarks of the same phenomena happening, and we just asked, could this be happening? And so again, with being at Mass General, that was literally walking down the hall to the brain bank, getting patients who had already donated, recruited by Merritt and James and everyone, and being able to test it relatively quickly. And there it was. And then from there, that's led to this discussion in the translational ALS meeting and to this clinical trial. So it's a journey, it's an odyssey.
It really is, and I think it highlights how much science can be not a straight line, and you have to sort of have your eyes open. And something that could have been regarded, truly could have just been regarded as an error, a mistake. Something happened to this mouse model, and we can't use it anymore, and there's nothing to be learned there. Instead, has really turned into something incredible. And we hear this again and again, but you have to be astute enough, and huge congratulations to you and your team to really be astute enough to see that, to ask the question, to be curious about it, and to follow it through to benefiting people. So thank you very much. Really excited about yet another trial that's going on now. And with that, we're going to turn to a whole different way of kind of looking at this disease, something that we really, I think, didn't even think was possible a few years ago, which is to think about how we might head off the disease. So the other half of the Mark duo here, Mark Garay, is going to talk to us about Prevent ALS, which is a really innovative project.
Great, thanks so much for the introduction, James. So I'm Mark Garay. I'm an instructor in neurology at the Healey Center for ALS at Mass General. And I'm really excited to talk to you about kind of our Prevent ALS study. This is really a pretty innovative study that is aiming to learn more about what do the earliest possible stages of ALS look like. And to do that, we're really studying kind of ALS gene carriers, like Mark was kind of alluding to. And we really hope that this study will eventually lay the groundwork to help both improve early diagnosis of ALS for both patients with familial and sporadic forms of ALS, and hopefully eventually allow us to help prevent it altogether. Next slide. Yep. So just to briefly review kind of the underlying epidemiology here. So you may know that most ALS is not inherited in families. In fact, 85 to 90% is inherited in what's called a sporadic fashion, where we have difficulty predicting who might get ALS or to another. However, 10 to 15% of ALS are caused by single genetic mutations or variants, as we prefer to call them nowadays. It's called a monogenic inheritance pattern. In many of these ALS patients, we see that they have a family history of either ALS or another disease called frontotemporal dementia. However, in some cases, this history is not elicited either because of a phenomenon called incomplete penetrance, where not everyone who has the mutation ends up developing ALS, or because people may die of other causes before ever developing disease. Fortunately, we've had tremendous advances in genetic sequencing technology over the last decade or so that have resulted in us being able to identify these gene variants for every ALS patient using free genetic panels that we routinely send from clinic. This can be done from blood or saliva. And so now the hope is that every ALS patient who has disease due to these mutations should be identified shortly after diagnosis of ALS. Unfortunately, the children and siblings of a person who has a familial form of ALS has about a 50% chance of inheriting the same genetic variant as their parent. These variants, if they possess them, put that individual at a very high risk of developing ALS at some point during their lifetime. We refer to these patients or these people who carry disease-causing variants but have not yet developed disease as gene carriers. Next slide. This slide just shows briefly kind of the tremendous advances we've made in terms of our understanding of the genetics of ALS. So since Bob Brown discovered the SOD1 or SOD1 gene as the first ALS disease-causing gene back in 1993, there has been rapid progress in our knowledge of these forms of familial ALS. And today we know of over 30 genes that can directly cause familial forms of ALS. And you can see that stark rise in the number of genes that have been described. The biggest circle right in the middle is the C9orf72 gene that, as Mark alluded to, is the most common inherited form of familial ALS, and it can also cause frontotemporal dementia. Next slide. Fortunately, we're now at a stage where, again, we've had tremendous advances in our ability to manipulate kind of the genetic sequences. And so in recent years, there's been a surge in development of therapies to treat familial forms of ALS. So as Merritt alluded to kind of during her talk, most recently, we were really thrilled last month in April when the FDA approved tofersen, or what's now known as Qalsody, which is an antisense oligonucleotide given via intrathecal or spinal fluid injection. This treatment is approved for ALS associated with SOD1 gene mutations, making this the very first approved therapy for a familial form of ALS. Critically, it's not just mutations, the SOD1 gene, that may be amenable to treatment. There are now other novel antisense oligonucleotide or gene therapeutic approaches that are currently in development for other familial forms of ALS, including due to mutations in the C9orf72, FUS, ataxin-2, or TARDP gene. A critical question, however, will be, when do we start these therapies? Rather than waiting for someone to develop disease, the question becomes, could we prevent ALS altogether by starting treatment in gene carriers who do not yet have symptoms of ALS? Next slide. So to help answer this question, so James and I, and with kind of the assistance of Katie Nicholson, who really started this study at Mass General, we've kind of formed a multi-center observational study called the Prevent ALS study. So this is a merger between an existing study called DIALS, that's a kind of collaboration between Mass General Hospital and Washington University in St. Louis, as well as another study called the ALS Families Project at Columbia University. Both of these studies are what are called observational research studies, where people enroll to kind of help us learn more about ALS as a disease, but don't directly offer a form of treatment like a clinical trial drug. These studies enroll family members of familial ALS patients or people who already know their gene carriers for a familial ALS gene. And what we do is we provide access to genetic testing with appropriate genetic counseling resources so that family members of familial ALS patients can learn about the risks of getting genetic testing themselves. These gene carriers who are at very high risk of developing ALS during their lifetime can really be a very critical window for us to learn a lot more about the early stages of ALS that is otherwise very difficult to discover or to study, since ALS is a very rare disease overall in the population. Next slide, please. So the principal goal of the Prevent ALS study is that we follow ALS gene carriers over time and really try to compare kind of multiple different tools that can be used to diagnose and follow ALS in its earliest stages. So a central part of this effort is we kind of perform blood and spinal fluid collections every 6 months to 1 year, as well as do other testing such as cognitive testing, quantitative strength testing, monitoring of patients' respiratory capacity. Other things that we're considering starting in the very near future would be using novel digital tools such as smartphone-based applications that could be used to monitor motor or cognitive performance remotely without someone ever coming into the office to be examined by a physician. While we're not performing this at MGH right now, the ALS Families Project at Columbia also performs regular EMG and nerve conduction studies to see could there be early changes on EMG that could predict the onset of disease before someone even realizes they have any symptoms related to ALS. Next slide. So since these studies began in 2017, initially as a small pilot study, we've really experienced very rapid growth in the number of participants who've been engaging in this research, despite really beginning large-scale enrollment in the midst of the COVID pandemic. So the table on the right is a little bit outdated, but now we have over 400 total participants across these across the Prevent study, with over 200 known gene carriers. About 85% of people who enroll in these studies elect to learn their genetic status, but about 15% opt to follow without learning their gene status, but want to just help our research. In addition to the SOD1 gene in addition to the C9orf72 that, as I said, was the most common overall genetic mutation, there are a wide variety of genetic variants that are represented across the study, and which has a lot of benefit to researchers who may be interested in learning more about these rare genetic forms of ALS. Next slide. And I think it's important to show how these studies are already providing crucial data for the design of ongoing ALS research studies. So a very similar study called to Prevent, called the PREFAL study at the University of Miami, recently demonstrated that rises in a protein called neurofilament light chain could be used to potentially detect the early changes in disease about 6 to 12 months before a gene carrier started to develop symptoms of ALS. The idea is this could represent a possible warning sign for ALS. Next slide. So right now, the PREVENT study is really aiming to kind of achieve similar goals. Could we find early warning signs for ALS in our gene carriers who we're following? We're currently working on evaluating whether we see the same rise in NFL prior to onset of ALS in our own disease cohort. One of the questions from the previous work that had not really been answered was whether mutation carriers, specifically of the C9 gene mutation, exhibit a similar rise in neurofilament before symptom onset, because the vast majority of patients in that prior graph carry mutations in the SOD1 gene. In addition, having blood and CSF available from a very large cohort of gene carriers also allows us the ability to evaluate potential new biomarkers that could facilitate, again, earlier diagnosis and potentially earlier treatments for ALS. So amongst that work, you saw some data from Mark Albers earlier looking at CCL2 levels within the spinal fluid of our C9orf72 gene carriers. That has really been facilitated by the work we're doing in PREVENT. But we're also working on other additional biomarkers, including levels of phosphorylated tau, as well as a new project evaluating something called cryptic exons that are reflective of changes in a protein called TDP43 that is thought to be critical in the development of ALS. Overall, we hope that these analyses will help our ability to detect early neurodegeneration in both familial and sporadic forms of ALS, and more rapidly start patients on highly effective therapies. The last thing that I didn't have slides for today, but I do want to just mention, is we now have really some of our very first research studies that exist to help us start to ask the question, could we prevent ALS? So Merritt had mentioned earlier that Qalsody or tofersen, which is the medication that was approved for the treatment of SOD1 ALS, has an ongoing prevention trial looking at potentially initiating treatment in pre-symptomatic SOD1 gene carriers who have a rise in neurofilament light chain levels. I'm currently acting as the site principal investigator for that clinical trial here at MGH, but it's a worldwide study. And so certainly anybody who has a known family history of familial ALS should very much consider kind of talking with a neurologist or talking with us anytime if they have questions about whether there could be preventative treatment trials for them as well. We're very excited about all of this work. Next slide. And with that, I just want to kind of acknowledge, our very large team. So specifically with the DIAL study at Mass General, Dr. Berry is the principal investigator for that trial. And then Katie Nicholson kind of helped start it before leaving MGH last year. And then we obviously have a great big team, including Diane Lucente, our amazing genetic counselor, and Praveen Pant, our nurse practitioner, and many research coordinators who help out with the study. And then we also have collaborators at WashU, Columbia University, as well as many basic science collaborators who we really all work together and we're very indebted to. So with that, I'll stop and ask for any questions.
Great, thank you so much, Mark. It really, just changes the way we think about this disease to be able to kind of approach the idea of preventing it. It really turns everything we do on its head, and it really is a different. We have to think differently when we're thinking about this population of gene carriers. There's one question that came in that I think is just about clearing up some of the specifics of the words. So I think you said 85 to of ALS is inherited in a sporadic fashion. And the question was, does that mean that it actually did occur in your ancestors, or so just maybe clarify that wording.
Yeah, so I would say that no, it doesn't necessarily mean that it did occur in your ancestors. What I would say is we typically think about sporadic ALS as ALS that occurs in an individual due to a combination of factors. So it's probably some combination of one's individual genetic makeup, so not just a single gene mutation, but multiple different genes that increase someone's risk of developing ALS, combined with things like environmental exposures or past traumas that could lead to the development of ALS in that individual.
So yeah, exactly. So sporadic.
I'm mostly misspeaking.
Well, right. I mean, sporadic ALS is one that is not passed down in a clear way from generation.
Correct.
So there's another question: is a 3-year-old genetic test that was done obsolete because of new information or new genes that are being tested for?
So I think it depends a little bit on that individual's kind of clinical and family history. So I think if there is a strong family history of ALS, we are learning about new genes all the time. And so it may be worth considering repeating genetic testing. And it's important to know as well that, there are kind of families that clearly have some form of familial mutation where multiple family members have been affected. And even today, our genetic testing is negative. And that may be because we haven't discovered the gene that is causing it yet, or there might be a technical reason why the genetic testing isn't quite picking up on it. I would say usually if someone has had negative genetic testing, maybe within the last couple of years, and has no family history of ALS, that's probably good enough. But it's always an important consideration and a discussion between you and your neurologist.
I think that's the key thing, because there are also different panels of genes, and they're kind of arranged in different ways, and even that's evolved over the last few years. Thank you very much. I'll point to one last comment, and then we'll take a break. So there's one other comment, and this is a new article by Stanley Prusner, who's a Nobel Prize winning researcher who discovered prion diseases, which are misfolded proteins that can show other proteins how to misfold as well, and is kind of the mechanism behind mad cow disease. And he published an article looking at the Parkinson's dementia ALS complex of Guam, and looking for prion-like proteins there. Fascinating read. This has been a phenomenon that's been very difficult to unravel. There was a very high incidence of this specific neurodegenerative disease that looks a little like Parkinson's, a little like a dementia, a little like ALS, and occurred in Guam, and then eventually sort of went away. And there's been different thoughts about why that was: environmental, genetic, prion. So really interesting article, and something that we all have our eyes on, because it could tell us about the pathology of neurodegeneration. So thank you for listening to that. If there are other questions for the panelists, we can certainly get to them kind of at the end of the time. If you think back on something and would like to clarify anything, please feel free to put it into the Q&A chat. We'll come back to it at an appropriate time. For now, we're going to take about a 5-minute break. We're running right on time, or maybe 2 minutes behind. And then we'll come back for our second session, which is innovation in clinical care. Thank you very much, everybody. Okay, I think it's time for us to get going again. And we'll talk about innovation in clinical care. Let's see. I think what I'm going to do is, so I'm going to talk about our approved drugs. And I think I'm going to try sharing my own screen. I'm going to ask for a little patience and a little help. So actually, Doreen, are you seeing the right? I'll just ask you, are you seeing the correct screen? Or are you seeing my?
No, correct.
Okay. All right. Very good. So I'm going to talk today about the FDA-approved disease-modifying therapies for ALS, which, until the last couple of years was a very, very short presentation where I just really needed one slide, and that's really different now. But I'm hoping to kind of walk through some of the mechanisms and some of the data behind the medications that many people with ALS are now taking. First thing to say is that we have 4 medications that are FDA approved to slow ALS progression. Plus, we have 1 FDA approved medication to treat specific symptoms. I'll mention that one. I'm not going to talk as much about it. But the first medication to slow ALS was FDA approved in 1995, and that's riluzole. This comes after really, hundreds of years of knowing about this disease, trying to understand it, trying to think about ways to treat it, and finally coming up with a treatment. That treatment is modestly effective, and we'll talk about it in a little more detail. Then in 2010, Nuedexta gained approval, and this is the drug that is not disease modifying. It was tested to see whether it slows the progression of the disease. It does not seem to do that, but it does have a remarkable effect on what we call pseudobulbar affect, which is this symptom where people can laugh more or cry more than their emotions mean. It's sort of almost like if you think of laughing or crying as a reflex, it's like too much of that reflex in a given situation. So people may watch a tearjerker commercial on TV, but have a difficult time stopping crying. And that can be bothersome, and this medication is remarkably good for that. Since then, there are also a couple of studies looking at the effect on speech and swallowing and finding some benefit for that medication. We may see more on that. In 2017, then, we saw the approval of Radicava or Edaravone to slow progression of ALS, at that point as an IV medication given 10 days out of the month. And then in 2022, an oral formulation was approved, and that is the formulation that we're most frequently prescribing at this point. The effect seems to be very similar, at least the dose that gets to the bloodstream seems to be very similar. That same year, just this last year, Relyvrio was approved in September. So 2 new drugs approved for ALS in the same year, really, really remarkable. And then just last week, Qalsody or tofersen gained approval for SOD1 ALS. So you can see the increased pace at which we're discovering and approving effective therapies, which is really remarkable for the field. So we now have these 4 FDA-approved disease-modifying therapies, and I think I'll just walk through each one to give you a sense of where we are with them. So riluzole was the first FDA-approved therapy. It slows disease by about 10%, and the original trial showed about a 3-month increased survival. That's maybe underestimating the effect. There have been some studies that show that. The formulation is as oral tablets, and they're taken twice daily. It's a well-tolerated medication. We do see some people who develop nausea at the onset of taking this. For most people, that will resolve within a week or 2. A few people, it's more severe, and we have to either lower the dose or take people off of the drug. And we just, one does that in a combination between the person with ALS and their clinician. Fatigue is listed as a side effect. This is a difficult one. Fatigue is a symptom of ALS. And so, it can be hard to sort that out, but, I think if people are experiencing potential side effects from a medication, they talk to their clinician and make a plan for how to figure out what the best way forward is. And then there can be effects on the liver, and as a result of that, they're very rare, especially at the dose that was approved, more common at higher doses. But we do blood tests for liver function every month in the beginning for the first 3 months or so, and then every 3 months after that up to a year, and then periodically just to keep an eye on it. So that's kind of the very practical part of it. So let's take a look at what the mechanism is. So riluzole does, we think, so first of all, I should say that for a lot of approved drugs, not just in ALS, but in general, we have a concept of what the mechanism is. And sometimes it turns out years later that we're not quite right about how they work. But what we think is that riluzole stops what we call excitotoxicity. And what that means is when one motor neuron becomes ill because of this disease that we call ALS, as it becomes ill, it gets hyperexcited and it fires too often, and it can overstimulate surrounding motor neurons. And what that means is that those surrounding motor neurons are then hyperexcited, and those neurons can then become ill, and they can overexcite the surrounding neurons, and there's this spreading of neuronal injury. So you can kind of see, if one neuron becomes ill, that neuron, begins to overfire, and then it can spread that sort of pathology or that problem to the next neuron that it connects with. And what riluzole does is come in and sort of in the middle and reduce the passage of that from one neuron to the other. And that's how we think that it's slowing the disease. We think that it is probably more effective to start it earlier. And so that's why this is always a part of the conversation very early and after diagnosis. Now, there have been a number of studies that look at riluzole. There were 2 seminal studies in the beginning that really set our understanding of it, but there have been a number since then. And then there has been what's called a meta-analysis done. And that means we look at many, many studies out there. And this combines 3 big studies. So the dashed lines are the studies that were looked at, and then the solid lines are the kind of combined analysis, which gives us more numbers. So those solid lines show you in red is the treated group and blue is the placebo group. And you can see that the progression is faster in the placebo group than the treated group. So that was our first kind of, approved drug. And that was all we had for a very long time. And now we have Radicava as well. So this is the second FDA-approved therapy for slowing ALS. It slows ALS by up to 30%. I'll show you some data about that in just a moment. It first came, as I said, as an IV infusion. It's now an oral gel. And the schedule is, it's taken 10 days out of the month. And that schedule is strange, but it comes from the fact that it was an IV infusion, which was much more complex to give. You heard Merritt allude to the fact that one of the ongoing trials right now is a daily dosing versus this kind of 10 days per month dosing to see whether one of those has a benefit over the other. It does not have many side effects. It tends to be very well tolerated. I have heard people say maybe fatigue, and that's listed. And then listed as a side effect is potentially difficulty with gait or difficulty walking. And again, I think if this is a concern, you just want to talk with your clinicians right away. There isn't any routine monitoring for labs that we need to do per se from a safety perspective. So Radicava works by reducing oxidative stress. This is something that we hear about a lot, oxidative stress, just in popular press and at the store when there are antioxidants that are being sold in the supplement aisle, but also people talk about blueberries as antioxidants. Oxidative stress results from really chemical reactions that are happening inside the cells, and those chemical reactions can damage the cells. Radicava is an antioxidant. So, if we think of oxidative stress as causing sort of these reactions going on in the cell and damaging that cell, then Radicava can come in and really sort of take away some of that oxidative stress and restore health to healthy neurons and make them less vulnerable to the disease. So that's how we think that this drug works. Again, there may be other mechanisms that it's working by. The initial study that was done with edaravone in 200 people with ALS came about 15 years after this drug was approved in Japan and Korea for treating stroke. So it was given to people after they had a stroke to reduce oxidative stress and improve outcomes longer term. It never got that approval in other countries, but they began to study it in ALS as well. And this first large study, unfortunately, didn't show an overall slowing of decline. But in certain subpopulations, they did see this, and they saw it more in people with definite ALS and people who were early, and that's by the Ellis-Scorial criteria. So what that means is 3 body regions were affected, as opposed to probable ALS by these Ellis-Scorial criteria. where 2 body regions are affected. So people who had more body regions affected and were earlier in the disease were the ones that seemed to have the most benefit. Now, it was not clear and remains somewhat unclear, but probably isn't that those people are different biologically, but rather that they're at a different stage in the disease. And indeed, these trials were 6 months long, and people who had change over those 6 months were more likely to have a detectable benefit of the drug. So the idea then from the investigators was to do a new trial and to sort of reduce some of the variability in the population. And indeed, in that new trial, they saw a 33% slowing of functional decline, as measured by the ALS Functional Rating Score, which is that 12-item questionnaire that many people are familiar with. Here we see in red the edaravone-treated group. Edaravone, by the way, is just another word for Radicava. They're interchangeable. One is the generic name, one is the brand name. And I'm sorry that we do that, but every drug has 2 names. So the edaravone or Radicava treated group is in red. They had a slower decline on the ALSFRS-R than the placebo group, and that works out to about 33%. This trial did not show a survival difference between those 2 groups. And so more recently, it was a part of the author group on this, we looked back at real world evidence. So this is coming from existing databases, large databases from medical records. And we found people who had been treated with IV edaravone or Radicava and people who had not been, all of whom had ALS. We matched them on their baseline characteristics. We did as careful a job as we could with that. And we found that there was a 6-month survival benefit for people who were taking Radicava. That's really the first time that we've seen that. There's another study that did not show a similar result. And just like riluzole, I think we're now having an increasing number of studies that are, after time, we'll sort of get a sense and be able to do some of these either meta-analyses or sort of get an overall sense of what is the magnitude of the benefit. That can take some time, but we guide ourselves by the data. The third drug that is approved for sporadic ALS was approved in September, again, based on a trial. Sabrina Paganoni was the PI of that, and the Healey Center was really the driver of that trial. Really an exciting thing that it was beneficial. So this is Relyvrio, and that has been shown to slow ALS by about 25% and then have a 4 to 10 month increased survival, and I'll show you where some of that uncertainty comes from and what we're doing to try to get a better idea of that. This is a powder. It comes as sort of a powder. It's mixed in water, 8 ounces of water. It's taken twice a day. One of the side effects is that it does not taste good. Taste is subjective, and it is really different from one person to another. And so, we get differing reports of whether it tastes bad or whether it is really, really difficult to tolerate because it tastes so bad. But nobody thinks it tastes great. GI upset occurs in some percentage of people. We're just getting a sense of that because it has not been on the market very long. But certainly some people have GI upset, usually lower GI, kind of abdominal discomfort. And some people have diarrhea with it as well. That can actually, both of those things, if they're mild, can resolve within a couple of weeks. Sometimes it needs, people need to adjust the way they're taking the medication. And again, if any of these things occur, you should talk with your clinician about it. There's no monitoring that needs to be done as far as labs or kind of keeping an eye out specifically on anything. It's usually pretty obvious if people have the GI upset. So Relyvrio is actually 2 drugs, and this comes back to that question of, what about combination therapies? Well, this was 2 drugs that were put together. The 2 people who started this company, actually looked at these mechanisms of the 2 drugs that come together and say, maybe they would actually benefit one another. Maybe they'd be better as a combination than alone. One of the drugs helps the cell produce energy at the level of the mitochondria, and the other helps manage the stress of misfolded proteins at the endoplasmic reticulum. Now, I don't need to tell you that it was at the endoplasmic reticulum, but that's a fun word to say. And many of us learned it in our 7th grade biology courses. So the endoplasmic reticulum is where we think about this stress. So if we think about this, it's sort of, the cell is lacking energy, and we have these proteins that are misfolded and building up in the cell, and things get messy, and the cell doesn't have the energy to fix it, and we get a downhill spiral. And that's why sort of attacking both of these things at once by giving more energy and a way to kind of clean things up, maybe helps the cell restore itself after becoming ill, or prevent a new motor neuron from becoming ill. And that's sort of the thought behind how this works. There probably are other mechanisms as well, because these are very complex drugs. What the trial showed was about a 25% slowing of functional decline. So again, drug in red, placebo in blue-green, and you can see that the slowing is the decline is slower in the treated group, and that was convincing. Then we looked at survival, and the survival data. So because of the design of the trial, the survival data is we have to sort of estimate. So we had a randomized trial for 6 months. Some people got placebo, some people got treatment. And after 6 months, everybody had the opportunity to go on the drug. So we can't compare people who, long-term, we can't compare people who never had the drug to people who did have the drug. We have to compare people who started the drug earlier to those who started later. And when we do that, we find a difference of, around 5-ish months. And But then we have some statistical methods that say, well, what if we imagine that people never actually started the drug? So let's sort of statistically model what might have happened if that placebo group had stayed a placebo group. And that gets us more towards sort of 10 months. And maybe even more when we look at historical controls. Now, this is really, that's wonderful data. We also would like to replicate it. So there is a trial going on now, and we had participants here in the US and at our center in the follow-up trial. It's called the Phoenix trial of Relyvrio. When Relyvrio was approved in the US, we transitioned everybody onto the actual open label commercial drug, but that trial continues on in Europe, and we should have results by this time next year from that follow-up trial. So we're eagerly awaiting that larger trial. And then the last approved drug is Qalsody, also known as tofersen. And this is approved for people with ALS due to a mutation in SOD1 only. And it has been shown to slow ALS and prolong survival. It's a sterile solution. It's given intrathecally. That means we do a lumbar puncture or a spinal tap, and we put it directly into the spinal fluid so that it's there bathing the nerves and the spinal cord and the brain. There are loading doses every other week, and then it's given monthly. There are side effects that we have to monitor clinically: headache, raised intracranial pressure, myelitis, aseptic meningitis. But these are things that we are learning how to monitor for, and they seem to be reversible when they occur. So tofersen is a part of a new class of medications that are called ASOs. And these can affect RNA and proteins in the cell in a very powerful way. In people with SOD1 mutations that cause ALS, SOD1 accumulates and causes the motor neuron to become ill and die. So it's that extra accumulation of SOD1 that seems to be the problem. We call that a toxic gain of function. Tofersen degrades the SOD1 RNA, and that reduces SOD1 and restores neuronal health. So if we look at this sort of in people with SOD1 ALS, that SOD1 accumulates and causes that neuron to die. But tofersen comes in and gets rid of that SOD1 and restores the health of the motor neuron. Now, in order to show you the results of this, I have to answer one more question, which is, what is neurofilament? Because this is key to how this drug got approved. And neurofilament, Marc Guéret talked about this as well. Neurofilament is a scaffolding protein. It maintains the neuron's axon. The axon is that long projection. Remember, motor neurons that live in our spinal cord send their projection all the way out to our muscles. They can be, 3 feet long. This very tiny projection called an axon, and it needs to be maintained. It needs something that gives it structure, and this is the protein that does that, neurofilament. So when a neuron is ill or dying, neurofilament is spilled from that neuron, from the inside of the neuron, into the spinal fluid and into the blood, where we can detect it as a biomarker. Neurofilament, or NF, goes up in ALS, and it stays elevated throughout the disease. You saw Marc Guéret show that it goes up probably prior to the onset of symptoms, and then it stays relatively stable. It can go up a little or down a little, but it's relatively stable. And you can see here in the panels labeled E and F that in people who don't have ALS, the levels are lower, and those with ALS, they're higher. And it seems to be higher in people who have faster progression. Here we show actually that it's highest in people with C9. That is not something that's actually borne out across studies. So I would say these probably were just fast progressors with C9. And then here we're showing neurofilament over time, and it's relatively stable. And there's a very small slope, and sometimes we see a small slope up, but it can go up or down. So in the trial of tofersen, we looked at SOD1 in the spinal fluid, in the CSF, and you can see in red, again, the treated group, the SOD1 comes down. This vertical line is when everybody started having actual drug, whether they were on placebo or not. And you can see then the people who had been on placebo, now their SOD1 comes down. And so the drug definitely reduces SOD1. That's what we expected. Neurofilament, again, the treated group, we see neurofilament coming down by 50%. Really remarkable effect. And then when the placebo group went onto the drug, you see their neurofilament coming down. This suggests to us that those neurons are not as ill. They're not spilling their contents into the blood and spinal fluid, and we're not detecting that protein. And that's really the basis of the approval of the drug. But we also see that the ALSFRS-R total score, while it wasn't statistically significantly different at the end of the randomized phase, those who got the drug earlier actually had a leveling out before those who got the drug later and did better. So we do want to get people on this drug early. Here we're looking at a measure of strength. And again, everybody declines, but boy, after 6 months, you see a leveling out. The people who got the placebo first continue to decline, and then they level out shortly after, 3 months after getting the drug. So really some evidence of benefit. And then here we're looking at the survival plots. It takes some time for those to separate. Luckily, we didn't see many survival events, that is, many deaths during the randomized trial. And actually, people are living long. What we know about natural history. So, I think we're entering a time of individualized or personalized therapy in ALS, and that's because we do have 4 drugs, but they're not all right for everybody. And so we see people, some of whom are taking all 4 of these, some may be taking 3, some are just taking 2 or 1, and not always the same one. And I think that tapestry is really incredible. To sort of see that we can design a program that's best for individual people based on effects, side effects, and practical considerations as well. So just to kind of round it out, we have 4 therapies that have been shown to slow ALS progression. 3 have been approved within the last 12 months. All can be taken together. Tofersen is only indicated for people with SOD1 mutations causing their ALS. Now, one last really important point here is that I think our practice has changed from genetic testing being interesting but not particularly important to really, I think all people should consider genetic testing with their clinician, because we do find people with SOD1 mutations that are unexpected, that look like they have sporadic ALS. And so it's become standard of care to at least offer this. People with ALS make personalized decisions about the right therapy plan with their clinical team. And really importantly, trials are guiding us to new, powerful, targeted treatments. Qalsody or tofersen is really the first in a new generation of powerful therapies. And I would not forget about multidisciplinary care, which is an important part of the care plan for people with ALS. Thank you.
Thank you so much, James, for that wonderful presentation. There have been some interesting questions coming in. So I was going to start with the first question. As many of you know, the American Academy of Neurology had its annual meeting in Boston in April, and a poster was presented that was entitled a 40-week phase 2B randomized multicenter double-blind placebo-controlled study evaluating the safety and efficacy of memantine in ALS. So the question here is, given that this was showing essentially a negative result, and it did not slow progression in ALS and also did not have an effect on some of the biomarkers, and that memantine is an NMDA antagonist and blocks glutamate, potentially, do these data de-emphasize the sort of glutamate excitotoxicity story in ALS? And furthermore, do you have any thoughts on why the results in terms of ribuzole and memantine are different? It's a great question.
It is a really good question. And this actually comes back to one of the comments that I made, which is that we sometimes think we know how a drug is working, and it turns out we don't know how a drug is working, because it is really true that if excitotoxicity is the mechanism for riluzole, and riluzole is really working by reducing that excitotoxicity, other drugs that reduce excitotoxicity should work. And we've been through now a number of other drugs, gabapentin and pyramate and amantine now, and many other drugs that should work, and they don't slow progression of disease. So there may be something different about either the way that riluzole is affecting excitotoxicity, or it may have a completely different property that we just haven't been focused on. And that there are lots of examples in medicine of that happening. Sort of as for sort of, where do we go now for something like memantine? I think that was pretty convincing negative evidence, unless there was some reason to think that the population wasn't the right population or the outcomes weren't right. But I think it was pretty convincing evidence that probably that drug won't, isn't worth pursuing further. Whether that means that the whole idea of excitotoxicity should be thrown out as a target, I don't know. I would say that the bulk of drugs that are being developed now are not focusing on excitotoxicity as kind of the target, because it's been challenging to find results there.
Thank you. The next question is about administration of Relyvrio. In this particular question, Marcus is asking, the instructions say to take it shortly before a snack or meal. In my experience, most instructions for medications say to take it either on an empty stomach or with food. Why can't this, why can't the medication be taken after or during eating a snack or a meal? And maybe more broadly speaking, maybe if you could also comment on sort of tips in administration, including like in regards to taste and.
Yeah, I'm sorry, this is for Relyvrio.
Relyvrio.
Yeah, Relyvrio, I mean, so one of the big challenges the two big challenges with Relyvrio that we're hearing from people are taste and, and stomach upset. Although I don't know always that the stomach upset is directly related to when it's taken. It's it can be separated by a number of hours. It doesn't need to be separated from food in the same way that riluzole is supposed to be separated from food. Although even there's some, you should talk with your clinician if you're having nausea with that one. Sometimes they have other thoughts about that. For the taste, people have tried a lot of different things. Someone recently told me that Hershey's chocolate sauce was actually cut the taste a lot, and then they went through a number of other kind of things they'd tried. Honey didn't do it, and so they, and some people will say that a little bit of milk shortly after it can help with the taste as well. Listerine for some people can help. Sometimes that just makes it worse. As far as taking it with food or not with food, it should not be put onto food. It shouldn't be mixed with milk, or which actually can make it taste better. But the problem is the proteins can potentially, we just don't know, bind some of the drug in Relyvrio. It shouldn't be taken with anything acidic because it will make the taste worse. So fruit juice would just make the taste worse from what we're told. The reason not to take it directly with food is that the idea is to try to give it a little separation from the potential other proteins that could bind some of the drug that you're taking.
A follow-up question about Relyvrio is also about sort of approval and access of the medication. My clinic is telling me that it's difficult to get Relyvrio approved for me on regular Medicare 4 years post-diagnosis. How do we deal with payers, and has CMS rolled on Relyvrio?
Yeah, so there's a process for whatever your insurance is. There's a process, and it's unfortunately it's often a longer process than any of us would like. There's a real urgency to getting people on these drugs. There are many steps that we have to go through, and I think that these companies are want to help and are working toward helping with that. It's a lot for clinic staff to do as well. What I would say about insurance and whether a given insurance will cover it is that there are so many different small differences that can make a big difference in terms of approval that I would stop short of saying anybody could or couldn't be approved. But I guess one doesn't know until they try in many ways. It is true that some insurances put parameters around how they will approve these drugs. So each of these drugs is approved. ALS broadly, tofersen is approved for SOD1 ALS. But the ones that are approved for ALS broadly, some payers are putting restrictions around them that people would have to qualify for the trial, or they have to have a vital capacity over a certain amount, or an ALS-FRSR. It's always worth pushing on. I think it's always worth pushing on those as much as possible, pushing back on the insurance. I don't know if others have different thoughts on that. I mean, Doreen, you deal with this a lot as well.
Mm-hmm. No, I
actually can say something else too. For Relyvrio specifically, I would recommend reaching out to the ACT program. They've probably already contacted you if your clinic is going through the prior authorization process. They're the people who work with Amylyx and the specialty pharmacies to get you the medication. They actually have a lot of grant programs that they've been working with so that they can help people get on Relyvrio, even if payers don't pay at all, or if they don't pay enough. And we have had a lot of success with those. Some of those grants close, but then they open again in a few days. So it is good to check back with the ACT program to hear about the grant programs, because even if your payer doesn't pay, you might still be able to find a way to get access to the drug.
Good, that's great. Yeah.
That's great. And then Dr. Berry, another question about adding or using vitamin B12 high dose in addition or in lieu of the FDA approved treatments. The question is that, the phase 3 trial in JAMA for 4 months had, showed pretty impressive data. So should B12 be considered early on along with FDA approved treatments, or in lieu of, or how do you sort of handle that?
Yeah, that's a great question. And I thought about whether I would include B12 in this presentation, and I decided that I would just focus on the ones that are FDA approved right now. So B12, just so to bring everybody into the question. So B12 in a form called methylcobalamin, which is just one form of B12, given at very high doses by intramuscular injection, was studied in a large phase 3 trial in Japan about 7 or 8 years ago, and the trial was negative. It didn't slow the disease. But in a subset of people who had disease less than a year, there was potentially a slowing effect. And so much like what happened with edaravone, the investigators took that subgroup and went and did a study in just that subgroup. So this is people within a year of the onset of their symptoms. And what they found in that follow-up study, which was only 4 months long, was that there was some slowing of decline on the ALS-FRSR in those who were getting B12. Now, B12 is a water-soluble vitamin, which means that if you get too much of it, your body uses what it needs, and then the rest, you pee the rest out, to be a little crass about it. That's different than some other vitamins. So the fat-soluble vitamins will build up and can build up to toxic levels, but B12 doesn't tend to do that. So treating with a very high dose seems to be safe, well tolerated. It's not FDA approved for this, but it is available through compounding pharmacies in some cases. And so it is something that we sometimes talk to people about. There is no conflict, no sort of interaction with any of the other approved drugs. So I don't think it has to be done instead of the other drugs. Cost is a factor. It's not wildly expensive, but it's not cheap either. And so people may be in a position of making decisions about what they, what they can afford. And I think that's a more complex conversation, and one that people should have with certainly with their physicians and their nurse practitioners to come to the right treatment program.
That's great. Thank you. One final question. This participant is asking, a blood test was done for the PMP program in 2016, came back negative, and then a second a couple months later. Is that of concern? Is it normal to not have any change happening, or how to interpret that? For a natural history study.
So I guess I'm not.
So perhaps, yeah.
I'm not sure what the blood test is. So let me give a couple of thoughts. I know the PMP program is doing some genetic testing. So if there's any uncertainty about genetics, it's important to talk to a genetic counselor and your treating physician, and maybe have, sometimes I'll pull in other geneticists or ALS geneticists to talk about complex results. We didn't talk about it a lot, but there can be some really complex results that come back from genetic testing. So if that's what it was, I would say take all the results, get all the results together, talk with your doc, see if there's a role for genetic counseling or ALS geneticists to kind of get involved, and you may need to get even a third test. What does change from time to time is what variants are called. There's something called a variant of uncertain significance, which means basically there's a change in your DNA. We don't know if that's just a change that we see all the time and it doesn't have any meaning, or gee, that could be a problem. So sometimes reanalysis is important. And that actually, there was a question earlier about having your genes retested. Sometimes you don't even need to have them retested. You can just have the company, the genetic testing company, kind of look at them again and reassess with new information about whether they're, whether it can resolve any questions. If this was about neurofilament, what I would say is, okay, I don't think PMP is testing neurofilament. We can now test neurofilament clinically. There's some variability in neurofilament, and we've seen this, and I've been testing it in some people just to get a sense of the test. I'm not really using it to learn about the disease so much as I am to learn about the test. I would say that and I've spoken with colleagues, both all of you on the phone and others. It's variable, and we're still looking for exactly how we use it clinically, I think.
Super. Thanks so much, Dr. Berry. We're going to move to our second speaker for this panel, which is who is nurse practitioner Jen Scalia, and she's going to be speaking about NGH clinical programs. Thank you.
Hi, I'm Julianne. Do you mind pulling up my slides? Thank you. This is a great place to start off. I think the last line of Dr. Berry's presentation was also ALS multidisciplinary care. And so that is what I am speaking about today. So next slide. So to start off right off the bat, the American Academy of Neurology actually just recommends multidisciplinary care. This is a formal recommendation. It's not really a nice to have. It's considered a need to have. And there's a lot that goes on in the ALS multidisciplinary clinic. There's also a lot that goes on outside of the ALS multidisciplinary clinic after you leave. So it's really good to have a time where you can come and see your doctor and nurse practitioner and nurse, PT, the whole team, so that they can have eyes on you and work with you to hear what your symptoms are, what's going on. And that way they can be this central spot for talking to lots of other people on your behalf. In clinic, we do a physical assessment and we also provide advice. And it's also getting to know you so that we can coordinate with, you can see kind of in this graphic how many people we can talk to about just one individual person and what their needs are. So getting to know you and what the right thing is for you so that we can talk to other people and advocate for you is really important. We also spend a great amount of time educating in clinic. Everybody is different, and there's a lot of information out there that may or may not be right for you. There's a lot of information on the internet. Some of it's true. Some of it's true for some people and not other people, and some of it's just not true. So trying to sort that out together is something we spend time in clinic doing. And then outside of clinic, which we've already touched a little bit on with the questions after Dr. Berry's talk, but we do a lot of insurance authorizations and advocacy. This used to not take up so much time because there wasn't as many medications. Glad to have that be the problem at that time because there's more medications available. Though it does take quite a bit of time, as I'm sure many people on this call are aware, because it's not that you leave clinic with a prescription and then you get to take the medication that night. There's lots of back and forth of sending notes, doing appeals, and the team is instrumental in doing that and advocating on your behalf. Next slide. So who do you come and see physically in clinic when you come every time? You see a physician or a nurse practitioner. We usually have a primary team method where you have both a physician and a nurse practitioner who know you, and you alternate who you see at which visit. The registered nurse is going to be the same that you see at almost every single visit, of course, unless they're out and somebody will be there to cover. But you will have the same MD, NP, and RN that knows you from day one onward and for forever. And they'll get to know you really well and work as a team to help you. We also have 3 physical therapists in our clinic, 2 speech and swallowing therapists, and they get to know you as well. Sometimes you alternate, though. They do try and stick with the same people so that you get to know one person, though it is nice to sometimes have someone's different point of view. So if you see one of our speech pathologists, Stacy, and the next one, Liz, sometimes they explain things in a slightly different way, and it's helpful to hear a different person say something in that different way. It might resonate a little bit better. We also do a breathing test of vital capacity at every single clinic visit, which is usually every 2 to 3 months. We see people in clinic. We do that to monitor how the muscles that help you breathe are doing. It's not a perfect method. It doesn't measure your muscles specifically, but it gives us a really good idea about how your breathing is doing. We have other breathing tests we can do depending on what your symptoms are, but we don't do those routinely, though they are available if we need to. We have 2 respiratory therapists now. They don't physically sit in clinic, so if you do know that you want to see them, letting us know ahead of time would be great, but they are in the hospital, and if you happen to be in clinic and a respiratory therapy needs come up, we can certainly page them, and they're happy to come and respond to you and answer your questions and sit with you. We also have a patient service coordinator. Her name is Larissa, and she sits in clinic to help with scheduling. We have heard that it can be difficult to schedule with us. We're working on that in a lot of different ways. But one of the ways that we are working on that is by having Larissa physically in clinic, so that ideally she can go in and see you, and you can walk out of the appointment with your next visit scheduled. So there's no phone tag or anything like that. We also have research coordinators. They're helping with many, many aspects of clinic. They're sometimes the ones who are bringing you to different rooms, sometimes the ones doing a breathing test or a questionnaire. They can also help with research questions. We don't do a lot of the treatment trials physically in clinic just because of space and time and resources. Sometimes you need different equipment, things like that. But we will sometimes do some studies that are just blood draw studies or filling out a questionnaire. And they're there and helping us to find out more information about ALS. I do also want to note that the physician and the nurse practitioner can both do virtual visits. So if there are times that you physically cannot come all the way into Boston, I get it. The traffic, the driving, all of it, it's a lot. We can do virtual visits as well. So there are some parameters around that now that the public health emergency has stopped. So we have to be licensed in the state that you're physically in. If I am personally not licensed in Connecticut, but if you drove a mile over the border into Massachusetts and sat in a Dunkin' Donuts parking lot and said, I'm in Massachusetts, then I could do a virtual visit with you. I know it sounds silly, but I just want to be very honest in case you don't live in Massachusetts, and that gets a little bit more difficult. It's also good to keep in mind that if you're traveling, you're in Florida on vacation, please know we are always here for you, but I can't physically do a virtual visit. You could always page me or any of your providers, and we can talk to you on the phone. We could talk to local providers. We do that all the time. We want to help do care, but physically seeing you on our screen is not something that we are legally allowed to do. Next slide. So those are the people who are available to you physically in clinic. We also have ALS specialists that are unique to our clinic, are known throughout the hospital as ALS, but are not physically in clinic and are available to talk to you. We have a pulmonologist. Her name is Kate Dudley, and she specializes in ALS and sleep medicine, actually. And she also sees patients in the respiratory unit in the hospital. So I'm hoping to not have to see any of you in the hospital as an inpatient, but if you are there, she is often one of the attendings on the respiratory floor. We have 2 genetic counselors that we work with. Mark Ray had mentioned Diane Lucenti earlier. She's involved in our research, and she does dabble in clinical care as well. We also have Helen, who does is more in the clinical side. And so they're more available virtually than in person, but we can make either work. And I should mention Dr. Dudley, the pulmonologist. She is available both physically and virtually, physically in person and virtually. It is helpful sometimes for her to see people in person, but because that's not always possible, she'll absolutely make it work for a virtual visit too. So we also have neuropalliative care. The doctor who is our neuropalliative care specialist is Dr. Kate Brizzi. I know it's Kate and Kate, it can get a little bit confusing, but Kate Brizzi is a neurologist, and she had a fellowship in palliative care. So she's a real expert at having conversations about symptom management specifically, and also talking about plans for the future. And so she's a really great resource. I think of her as a sounding board. We also have an occupational therapist, Julie McLean, and she's available by appointment only. She's in the same building as our Wang Boston clinic, but just not physically in our space. And we also have a dietitian now. Her name is Jamie Gary, and she's available by phone call. We let her know if you're wanting to talk to her, and then we can let her know to give you a call, and she'll talk to you on the phone about some of your dietary needs. We have orthotists, a few that we work with, and we also have a research access nurse named Judy Carey, who has been mentioned a couple of times already today. And sometimes she's physically in clinic, but more often than not, she's doing video or phone calls. Either way, we really think of research as a part of our clinical care. And when people are in research, we can't not take off our clinical hat, and we're thinking clinically too. It's incredibly integrated. And so Judy is absolutely considered a member of our clinic team. Next slide. Of different programs. One is called Parenting at a Challenging Time, or PACT, P-A-C-T. And this is run by Archana and Cindy, and they're child psychologists, and they provide guidance to parents or grandparents about how to talk to their kids about their symptoms, their medical decisions, or ALS. They may have some recommendations about how to talk about it, but if that's not right for your family, they are more than happy to work around what's right for your family. But it's really helpful to have two trained psychologists tell you, developmentally what to expect with the kids in your family, and how to talk to them about ALS, who's going to be online Googling, who's going to show, coping in different ways. And they can be such a tremendous resource. This program came out of a cancer program doing the same thing of how to talk to kids about cancer. And we're very grateful for them to be involved in our clinic. You can meet them once or multiple times, and they don't talk to the kids. They just talk to the parents about how to talk to their kids. Next slide. We also have a house call program. In the big picture here, this is the originator, the very first nurse in our house call program. Her name is Katie T. She since has left to go to school to be a nurse practitioner. But the other two photos is Carl, who's an NP, and Kristen Kingsley, who is an RN, and they will physically go to your house if you are in Massachusetts, roughly east of 495, just because there's only two people, so we can't quite go much further at this point in time, but they will physically go to your house to see you in your home. And it's like bringing a piece of the ALS clinic to your home. It's not the full multidisciplinary clinic, but it is an ALS specialist, someone who's part of our clinic, and it's going to see you in your home, where it might be a little bit more relaxing. They can also see the environment that you're in. And it's also just in general, a great touch point to have in addition to your clinic visits. We do this program in collaboration with Compassionate Care ALS, which some people are already a part of, and then they join the house call program, or vice versa, but we do it with them. They're a nonprofit who are knowledgeable about ALS, and they're a great companion to have in these visits. Next. And I just wanted to mention that we have a new Waltham location. It just started this past Monday, May 1st. So now your full multidisciplinary clinic is in Waltham, which is easier for some people's drives. If it's not easier for you, we're not going anywhere. We're very much in Boston, but we are trying to, be available to everybody and anybody. And it's, for some people, makes a lot more sense to be going to Waltham. Sometimes it's the commute, sometimes it's the comfort driving in Boston, whatever it is. But this is a new service we have, and that you get the same exact people in clinic as in Boston. You get that in Waltham too. And next, I think that should be it. Yep. And that's all I have for now. I am looking at the time, so I don't know, Doreen, are we going to wait for questions until after Suma or now?
We'll hand this over to Suma Babu,
Okay.
who's going to be presenting on expanded access programs, and we'll save all the questions to the very end.
Sure.
Thank you so much.
Can everybody see my slides okay? To present to review. Everyone, so last talk of the session. I think throughout this session, we've heard so many high notes about research, therapies, and care innovations in ALS. And we're really at an exciting time that we're seeing so many new things happening, all in the right direction. To help and benefit our patients. And today I'm going to talk about another topic that is rapidly evolving, the topic of expanded access protocols in ALS. And I hope that I'm able to end this session on yet another high note as well. So what are expanded access protocols? Some people call this as compassionate use protocols. So expanded access is primarily meant to provide access of an experimental medication to patients who otherwise do not have an opportunity to participate in research trials or get access to experimental medications. And there are some very specific definitions and recommendations from FDA as well about expanded access protocols and approaches. And the fundamental important point to consider is that this is primarily meant for patients who otherwise don't have an opportunity to participate in traditional clinical trials. And another important point that FDA actually stresses upon is that these expanded access protocols should not interfere with the completion of the traditional clinical effectiveness trial, which is what is needed, towards bringing a drug towards that finish line of getting that FDA approval as a standard of care therapy for ALS. So expanded access protocols can run in parallel to traditional clinical effectiveness trials. And in fact, actually, FDA encourages companies or industries that are developing drugs for ALS, specifically for ALS, to incorporate expanded access programs or protocols during their drug development. And this is the exact verbatim term from the FDA guidance to industry. They say that whenever possible, data from patients who may not have been included in effectiveness studies, but in whom, based on other data, the use of the drug following approval is likely. So this is essentially, encouraging companies to use EAPs or expanded access protocols to collect long-term safety data. So it's not just providing access to an experimental medication, but also gathering data about safety in a population that otherwise doesn't participate in clinical trials. So there are some criteria that we think about while developing or expanded access protocols. Right now, this is an evolving space. So typically the way the expanded access programs are working are that, companies or industries that are developing drugs for ALS may set aside a small amount of the drug to donate to patients who are otherwise not eligible for trials. So the number of spots available is definitely very small compared to the large demand that we currently see in the ALS community in terms of needing access to experimental medications. But this is just the start of a new era of trying to get access to research and experimental medications. So this is continuing to grow and expand, which I'm going to talk about in just a little bit. So typically, in order to qualify for an expanded access protocol, there are some definitions according to FDA that a patient has a life-threatening condition or a serious condition, and there's no similar, it has to be no, I'm sorry, it's the typo there, no similar or satisfactory alternative therapy, and enrollment in a clinical trial is not possible. And because it is still experimental, we still have to keep in mind about the risk-benefit ratio, and there should be good justification to say that the possible benefits actually justifies the possible risks of treatment. And patient who's taking the investigational product will not interfere or affect with the original clinical investigational clinical trial itself. Generally, when we're developing expanded access protocols, there are some design differences compared to traditional clinical trials. Expanded access protocols can be for a single patient or a single patient IND EAPs, they're called as, or it could be a cohort of patients. It could be at one site or it could be at multiple sites. And generally, because this is targeting patients who don't meet the clinical trial eligibility criteria, some of them may be further advanced in their disease journey, or some of them may have a very slow progressing disease that many of the traditional clinical trials may exclude from their trials. So the eligibility criteria for EAPs tend to be broad and more inclusive. And there's no placebo in EAPs as such. And there could be a lot of flexibility with study visits as well. And it really depends on the EAP, whether you can have broad visit windows, or the visit windows have to be predefined and established based off of what is expected from that experimental therapy. Some EAPs can be run completely remotely or virtually, but others may still require you to come into your site or for infusions and other things. And some EAPs may run long-term, depending on the drug availability and funding resources. And this model has been successful in cancer studies as well, where EAPs can run indefinitely as compassionate use protocols. And we're starting to see the start of EAPs in ALS. We're not quite there yet to run EAPs indefinitely at the moment, but that's the future that we're looking forward to as well, as that every ALS patient has access to experimental medications, either through clinical trials or through an alternate pathway, such as an EAP. And most industry-sponsored EAPs may think about establishing expanded access protocols at a time in their drug development where they've gathered sufficient safety data about their drug, especially given that many of these patients could be further along in the disease journey, as well as there is you could potentially run these trials remotely. We want to make sure that the drug that we're giving is safe, and even having the flexibility of study visits, we are able to maintain good oversight of the expanded access protocol without compromising on the safety. So if a drug is in an early phase development, sometimes EAPs may not be available, and it may be available only for later phase development as such. At MGH, we tend to think about EAP as an extension of clinical care. And so on one hand, you have the clinical care domain, and on the other hand, you have the research model. So where we integrate the two together, we can look at these two different domains as one big single entity. On the clinical side, we try to align EAP study visits along with the clinical visits with your nurse practitioner or your ALS neurologist. And we try to incorporate some of the clinical and research staffing resources we have at MGH Clinic, as well as using some clinic spaces as well for some of the study visits. And some institutions may even use your electronic medical record as your source document for collecting and gathering data for the EAP. On the research side, despite this being as an extension of clinical care, it is still considered as research. There's still a research protocol we have to follow. There are still some regulatory requirements. We need to get approvals from IRB, from FDA, and we need to make sure that the industry sponsor is able to provide the drug. And there is, again, that risk-benefit discussion that needs to happen. And I think it's very important for patients to understand what you're signing up for, what the risks are, what the benefits are before you enter the study. And if there are any side effects that happen during participation in the EAP, all of that needs to be reported appropriately in a timely manner to several regulatory agencies, including the IRB, the FDA, and the sponsor as well. So in terms of the operational structure, I'll show you in just a little bit about how these EAPs are evolving and how the complexity of running these EAPs are also evolving in parallel. If you're dealing with a single site or a single patient EAP, this is sort of what the ecosystem looks like. At the center of this ecosystem is really the patient. And if a patient qualifies for an expanded access protocol, the patient and their treating provider will then reach out to the industry that is running or developing a drug and see if they're able to provide drug for an expanded access protocol. If the company agrees, then you get approvals from the Institutional Ethics Board or IRBs, and then you go to the FDA, get approvals, and you create a protocol. You tell all the parties what you're going to do in this research protocol and how you're going to treat the patient, how frequently, what dosage, all of these things, and then you run the study. Now, we are entering this era of multi-center expanded access protocols. Our goal is to really broaden the access to experimental medications for patients beyond MGH, beyond a single site, so that people can get access no matter where they are in different parts of the country. And the very first multi-center expanded access protocol was developed as a companion to the HEALEY ALS Platform Trial that you heard about earlier in the session. So to date, we have activated 10 sites, and about 81 expanded access protocol participants have enrolled across these 10 sites. And in this model, the company typically provides the drug, and the rest of the operational costs are all undertaken, covered by either philanthropic resources or foundation. And here, the regulatory responsibility is maintained by the company that is developing the drug, but also the IND is held in some cases by a Healey Center, MGH Healey Center investigator. And in addition to the companion to the platform trial, there is yet another multi-center expanded access protocol. This is the first international expanded access protocol. This was developed by Biogen, and this is for tofersen, which was just approved in the past week, which is very, very exciting for SOD1 genetic form of ALS. And in this program, there are about 120 sites worldwide, and more than 300 participants have enrolled in this EAP. And we were fortunate to be one of the sites as well. And even before this expanded access protocol took off from ground, we had a single patient EAP that we were able to offer to several of our SOD1 ALS patients at MGH. And in this model, the industry provides the drug. And this is a little bit more complex model because it involves lumbar punctures, infusions, long study visits, frequent study visits on site, and you need a research space where these study procedures are conducted. So the company provides the drug, but all other operational costs are undertaken by sites using their philanthropic or foundational resources, and so on and so forth. And some sites have been also maybe billing the patient's insurance for some of the safety labs and procedural costs and things like that. Any safety signals that are emerging from the expanded access protocol, as I mentioned before, are being reported in a timely manner to the FDA and to the sponsor as well. So in December of 2021, there was a law that was signed, there was a bill that was signed into law called as the Act for ALS. And this really has opened up a new era of expanded access opportunities for ALS patients. And under this law, the National Institutes of Health is able to actually support and sponsor expanded access protocols, not only for the purpose of providing access, but also to conduct clinical research to try to understand safety as well as effectiveness in the population that is eligible for EAPs and are not eligible for traditional clinical trials. So this is the first time that this is happening in ALS, which is very exciting. And so essentially any data that is gathered from the NIH-sponsored EAPs would potentially serve both from safety as well as effectiveness towards that final package when the drug development continues, and hopefully one day it goes towards the finish line of submitting for FDA approval, then this data would also support that drug development at that time point. And we are very fortunate that Dr. Paganoni, Dr. Berry, and I have had the opportunity to receive the first grant. The MGH Healey Center has received the first grant under this Act for ALS, and we currently have an ongoing multi-center expanded access protocol for Trehalose IV infusion. And we have activated a few sites in this expanded access protocol. And just in the past 2 months, in March of this year, we enrolled our first participant. And this study continues on, and our hope is, and our target enrollment is 70 participants across 25 sites in the US. So very, very exciting. And earlier I showed you what a single center EAP model looks like. And now as you grow the EAPs and we expand into multi-center EAPs, the complexity of organization operation grows many fold. And this is sort of what it looks like in terms of the EAP overview. So in addition to MGH, now we have to get approvals from all 25 sites, their ethics board, as well as, there are multiple vendors to help with the weekly infusions of trehalose, as well as making sure that every site, every patient, no matter where they are in the US who are participating in the trial, get access, that the drug gets shipped to their location in a timely manner, and they're connected with all the resources to conduct the study, collect the data, and making sure the data is all clean, that we can actually then analyze and involve the patient community and have good regulatory oversight over the study and report whatever data is collected from the study to the sponsor, that is Healey Therapeutics, to NIH, as well as ultimately to FDA. And then if you zoom in and look at what the MGH team looks like, there again gets more complex. With the MGH team includes not only the investigators who are leading the study, but also a large group of a large team that is actually involved in project management, grants administration, quality assurance, data management, and systems. You look at each individual site that's participating in the study, as well as vendors. There are a large number of vendors who are making this EAP possible. And at each site, there is, again, a large group of people that are involved in making this EAP happen, including nurses, coordinators, grant administrators, clinicians, as well as additional site resources. So, any project that has to evolve and have a large impact and have a large outreach operationally becomes complex. But we are at a very, very exciting time that we're able to offer this, and it is just a start of a new era. Just to summarize, at MGH, when we started the EAP program about 5 years ago, with the vision of Dr. Sukovich, we started the first EAP. And over the 5 years, this program has grown into multiple programs, multiple patients who have had the opportunity to participate in this. And I want to give a special shout out to Sarah Lupino, who couldn't be here for the session today, and she's been spearheading and leading a number of these single patient IND EAPs, as well as initial cohort-based EAPs, in terms of getting them off the ground and running the trials and really expanding this program. So now to date, we've had 27 separate EAPs, and we currently have 14 active EAPs, and over 240 participants have enrolled since 2018. So it's all very, very exciting to share with you today. So if you're interested in participating in EAPs, and if you do not qualify for other clinical trials, this is a link to find more information. You can also talk to your neurologist as a first step to see if you qualify for EAPs and if there are sites closer to where you are as well. So with that, I will stop, and I'll also stop sharing. Thank you. Is Doreen here?
Thank you so much, Suma. It's so exciting. I wasn't sure, Dr. Berry, if you wanted to finish with closing remarks. The only additional question that came in was probably for Jen Scalia. And it's about, I basically asked, neurogenic bladder and bowel, sort of how is that addressed in the clinic? Classically in ALS, the bowel and bladder function are not affected. And so rarely it could be. And I guess the question is, if the subspecialty is not reflected in the clinic, how do you sort of coordinate that care?
Sure, yeah, as you said, Doreen, it's not that all patients with ALS experience bowel and bladder problems, but I will say it does become a big topic because a lot can contribute to bowel and bladder problems. Like if it's physically more difficult for you to get to the bathroom, then people might find that they are urinating prematurely, having accidents, and it causes a lot of difficulty. It's not always neurogenic bladder that's causing that though. So it's really good to talk to your clinical team because it might be something that your ALS team can help you address, or at least do a few things to sort out first. Also always loop in your PCP, because it could sometimes be UTI. But if it is something a little bit more difficult to diagnose and to really get to the bottom of, we always talk to other providers. So at Mass General, there is a urologist that we've actually been using for a few patients, and we've been collaborating with him. If it's not him, his name is Pablo Gomari, then we will talk to anybody, any urologist, any GI specialist that wants to help sort this out with us, and we can give them our information about ALS, and then we utilize their information about urology or GI, and we sort it out together. So there's lots of coordination that goes on in the background.
I would just add a small plug in there also for about a survey study that we are currently running to understand the impact of bladder disturbances in ALS. So if anybody's interested, it's on the Healey Center website, and you could also reach out to me. I'm happy to share the link as well.
Yeah, so and I think it'll really give us new insights, because it is something, as you guys are both saying, that we see, we manage, we deal with. How frequent and what the etiology is, not we haven't written it in the books yet, so we're often just sort of feeling it through. So it'll be great to have this data. Look, I want to thank everyone on our team for being here, but all of our guests, all of the people in our community who joined, everybody who listens to the recording. I hope that this gives a little taste of how rapidly things are evolving in the care we give, in the way we think about that care. We're rethinking, as you just heard in that conversation, we're rethinking some of the very most basic things we think we know about this disease. And we are seeing new drugs be approved by the FDA, and we have more trials going on than ever before. These are real, tangible changes, and we are so proud at the Healey and AMG Center for being able to have some role in moving that forward. And we are so thankful to everybody in the clinic, in our trials, in the community who is lending support to that. So we hope that this was informative and fun, and we look forward to doing more of these. Thank you very much. Thanks, everyone.